Hello everyone, welcome back. So this video is about second week of development. So in the first week we saw that there is inner cell and outer cell formation. So inner cell will be becoming the future embryo proper and the outer cell mass will become the trophoblast. So let's look into what is happening in the second week of development. So during second week of development, the inner cell mass will form the embryoblast, and this embryoblast will form two layers. So this embryoblast will form the epiblast layer as well as the hypoblast layer. So this epiblast cells will be columnar cells, and the hypoblast cells will be cuboidal cells. And this epiblast will be forming on the dorsal end, whereas hypoblast on the ventral end. So basically this will be the epiblast layer. It will be columnar cells, and in the ventral end we will have the hypoblast cell which will be cuboidal. So this epiblast cells will enclose the amniotic cavity, whereas the hypoblast cells will form a membrane. So hypoblast cells, which is cuboidal cells on the ventral end, will form a membrane which is called Hcg membrane or the exocellular membrane. So the hypoblast cells will form exocellular membrane. It is also called Hcg membrane. So this membrane is made up of flattened cells, and thus this hypoblast will enclose the primary yolk sac. So the amniotic cavity will be enclosed by the epiblast, and the primary yolk sac will be enclosed by hypoblast. So if we take an ultrasound in the second week of development, we can see in ultrasound the double bleb sign, and this double bleb sign is seen because, as I said, we have the formation of amniotic cavity and the formation of yolk sac. So in ultrasound it is seen as double bleb sign. And this was about the inner cell mass. Inner cell mass, epiblast cell forming the amniotic cavity, and the hypoblast cell forming the primary yolk sac. So what about the outer cell mass? We said in previous videos, like outer cell mass will form the trophoblast. So trophoblast was formed on the first week of development. So in the second week, that is specifically on the 8th day, the trophoblast is divided into two layers. So we have the cytotrophoblast and the syncytial trophoblast formation on the 8th day of development. So cytotrophoblast, as like the cells will have a single nucleus and the membrane is well defined membrane, whereas syncytial trophoblast, as the name suggests, it is syncytium, so cells are like multinucleated and there is no well defined membrane. So in this diagram we can see the primitive yolk sac or the primary yolk sac, which is formed by the hypoblast and the Hcg membrane, and this is amnion and the amniotic cavity, which is formed by the epiblast and the trophoblast. So we can see the cytotrophoblast. So it is single nucleated and well defined membrane, and this is syncytial trophoblast, which is multinucleated and there is no well defined membrane. So let's look into the formation of uteroplacental and fetoplacental circulation. So basically in the syncytial trophoblast, lacunas are formed. So syncytial trophoblast, which is outer layer, which is multinucleated and not well defined. So there is lacunas formed in the syncytial trophoblast layer. So these lacunas are filled with bloods, and it erodes the endometrium and vessels. So the blood from the endometrium, so the lacunas are filled with blood and so this blood will then the uterine or the maternal vessels blood is filled in the lacuna, and this blood through diffusion it reaches the inner cell mass and thereby supplies the inner cell mass, and thereby the embryo is getting blood through this lacuna by diffusion. So thus the uteroplacental circulation is established. So this uteroplacental circulation is a primitive type of circulation. It is not enough for the embryo to grow properly, because embryo needs enormous amount of blood, and through uteroplacental circulation it is getting a limited amount of blood just through diffusion. So this uteroplacental circulation is formed on the 15th day of development. Previously it was said like it was on 12th day. Now the latest edition it is 15th day of development. So the if 15 and 12 is given, 15th day should be preferred over 12th day. And the uteroplacental circulation will be replaced by the fetoplacental circulation. As I said, because of the enormous amount of blood requirement of the embryo, the primitive hard tube will be formed, and that will replace the uteroplacental circulation into fetoplacental circulation. So this fetoplacental circulation is formed on the 17th day. Early it was 22nd day. So recently it is said to be 17th day fetoplacental circulation is formed. So hard tube will be formed, and which maintains this fetoplacental circulation. So in this diagram we can see the syncytial trophoblast layer, the cytotrophoblast layer, and the blood filled lacuna in the syncytial trophoblast, through which the uteroplacental circulation blood reaches the inner cell mass. So here in this diagram we can see the extraembryonic mesoderm layer. So this layer is happening between the inner cell mass and the outer cell mass. So if we get a question like extraembryonic mesoderm is derived from, then it will be from the primary yolk sac only this extraembryonic mesoderm layer is derived. So like in option, if it is like epiblast, hypoblast, or primary yolk sac, the extraembryonic mesoderm, the right answer will be primary yolk sac. So now let's see what is chorion and the chorionic villi formation. So chorion is basically made up of three structures. So it is extraembryonic mesoderm, the cytotrophoblast, and syncytial trophoblast. So this three component forms the chorion, and the primary chorionic villi is formed when the cytotrophoblast invades into a syncytial trophoblast. Then we get the primary chorionic villi, and in primary chorionic villi itself, if there is mesoderm core invasion, like this extraembryonic mesoderm also gets inside the primary chorionic villi, then it is secondary chorionic villi formation. So basically chorion is made up of extraembryonic mesoderm, cytotrophoblast, and syncytial trophoblast. The primary chorionic villi is formed when the cytotrophoblast invades into the syncytial trophoblast, and secondary chorionic villi is formed by cytotrophoblast invading to syncytial trophoblast plus the extraembryonic mesoderm inversion as well. So that was about chorion, primary chorionic villi, and the secondary chorionic villi. So now I'll talk about the secondary yolk sac and the umbilical cord. So as I said before, there is extraembryonic mesoderm, the yellow color in the diagram. So in that, cavities are formed, and the small cavities formed in the extraembryonic mesoderm, it will join to form a larger cavity. So in the extraembryonic mesoderm, where the cavity is formed, and the part where the cavity is not formed, which is the undivided part of extraembryonic mesoderm, which is cranial to the amniotic cavity, so that part of extraembryonic mesoderm will be forming the connecting stalk, and this connecting stalk will become the umbilical cord. So the cavity inside the extraembryonic mesoderm, it is also divided into two. Like there is plagnopluric layer of extraembryonic mesoderm, which is also called the visceral layer. So it will be surrounding the yolk sac, whereas and this the yolk sac will be made smaller. So then when this plagnopluric layer is formed over the yolk sac, then that yolk sac will be called secondary yolk sac. So the difference between primary and secondary yolk sac is that the secondary yolk sac will be smaller in size. So in this diagram it will be clear, like this yellow color is the extraembryonic mesoderm. So the mesoderm, there is cavity formed, that is a green color, and this cavity which is on the upper part, like upper part of extraembryonic mesoderm, that is this part. So it is an undivided part of extraembryonic mesoderm, which is cranial to the amniotic cavity. So it will form the connecting stalk and it will form the umbilical cord. Whereas because of the cavity formation in the extraembryonic mesoderm, so we have this part of mesoderm which is called plagnopluric layer of extraembryonic mesoderm. So basically the blue color, so it is covering the yolk sac, and all the rest of the part of extraembryonic mesoderm, it is called somatic layer of extraembryonic mesoderm. So the extraembryonic mesoderm is divided into two, which is plagnopluric layer or the visceral layer and the somatic layer. So the amniotic cavity, which is this cavity, so the inner surface will be formed by the amniogenic cells. And if we get a question like what is formed by the outer surface of the amniotic cavity, so it will be this layer, which is the somatic layer of extraembryonic mesoderm. So that was about the second week of development. So I would say I'd like to say the summary of second week of development. So basically in second week of development, all two things are formed, like inner cell mass, two layers of inner cell mass is formed. So that is hypoblast and the epiblast. So and two layers of outer cell mass is formed, which is cytotrophoblast and syncytial trophoblast. And the amnion and the chorion are formed. Amnion, which is derived from the epiblast, and the chorion, like three things, like which is extraembryonic mesoderm, cytotrophoblast and syncytial trophoblast. And also extraembryonic mesoderm, two layers are formed, which is somatic and the visceral or plagnopluric layer. The yolk sac is also divided into primary and secondary yolk sac. So again in the diagram we can see extraembryonic coelom, the cavity, and the extraembryonic mesoderm covering the yolk sac, which is plagnopluric mesoderm, and the rest part, which is called somatopluric mesoderm, and amniotic cavity, which is covered in inner surface by the amniogenic cells. And we have this epiblast and hypoblast layer, hypoblast then enclosing the primary primitive or the primary yolk sac, and the epiblast forming the amniotic cavity, and we have the cytotrophoblast layer. So now in applied, where we talk about Hcg hormone. So I'll say I mentioned about Hcg in the previous video, like Hcg is secreted by which layer of trophoblast? By the syncytial trophoblast. So Hcg basically induces the hormone progesterone, and progesterone helps in the maintaining the pregnancy. So the question arises when we can find out the level of Hcg. So on the 8th day in maternal blood, Hcg can be measured, whereas on 10th day in the maternal urine, the Hcg can be measured. Maternal urine, the Hcg can be measured. So because Hcg can be measured in maternal urine on the 10th day, Hcg assay can be done like on the first day of mixed period. but those sample of urine should be on the early morning sample. Hcg can be measured. So basically, if a female, she had ovulation on like 14th of a month, and she had to get her periods like on March 1st, and she is not getting her periods, and if she checks the level of hcg in her urine, and it can be found out if she is pregnant or not, but because uh, because in the first day of missed period, it's 100% sure that hcg levels can be found out. So there are some conditions where very low hcg levels will be there. One such condition is ectopic pregnancy, because the implantation is taking place not in the uterine cavity. Because of that, the syncytial trophoblast layer will not be well formed, and this syncytial trophoblast is the layer which is producing hcg. So in this case, hcg will be low. And also in case of spontaneous abortion, some cases where there is high hcg, like in multiple pregnancy, in molar pregnancy, also in some choriocarcinomas, and this gestational trophoblastic neoplasia, that is cancer. In such cases, we have high hcg levels. So that is the end of this video of second week of development. Thanks for watching the video. Please like, share and subscribe. Thank you.